Hi, my name is Cora and I'll be talking to you about chromosomes during meiosis. During meiosis, chromosomes with two chromatids will condense and recombination may occur. In this example, two homologous chromosomes of individual 1 are pictured, with the letters representing regions of genes. First, we need to determine if there's an aberration in the chromosomes. By looking at the order of the areas, we can see IJKL has been inverted. Since the centromere is located between the J and K, which is inverted, we know that the chromosome underwent a pericentric inversion compared to the original. In this model, the white chromatids are in the original order while the yellow are the inverted chromatids. The gene regions are unable to pair, so in order to become aligned, the inverted chromosomes will create a loop. While in this loop, it is possible to have crossing over events, such as one between the J and K locus. The simplest chromatids to determine the regions of are the non-recombinants, starting with the original chromosome. Then we can follow the inverted chromosome not involved with the crossover. Here is a simplified version of the model with the non-recombinants removed. Starting from the left of the original chromatid, the first portion will be GHIJ, but after the crossover point, it will be KLHG. Then we can move to the other side of the original, producing a chromatid with the regions MLKJIM. For the first two non-recombinants, it will have every gene region and a centromere, so are therefore viable gametes. For the bottom two chromatids, they have repeated regions producing excess gene products and are lacking in others, resulting in inviable gametes. In another example, the two homologous chromosomes of individual 2 are shown. When looking at the 4 chromatids, the gene regions are able to pair directly with each other, unlike what happened with individual 1, as the chromosomes are identical in order. With the crossover event between the J and K locus, we can track the 4 chromatids produced. There will be chromatids produced that are non-recombinant, such as by following the top chromatid and the bottommost chromatid. By following the second chromatid of the first chromosome, we end up with a chromatid that is half the first and half the second. Then, by going the other direction of the second chromatid on the first chromosome, we also end up with a mixed chromosome. Overall, the 4 gametes will have one of every gene's region and a centromere, meaning that all 4 gametes produced will be viable. When comparing individual 1 with individual 2, the former only has 50% viable gametes, while the latter has 100% viable. This means that the fitness of individual 2 will be greater than individual 1, as individual 2 produces twice as many gametes that can develop into functional organisms.